And John tried to, he had put out notifications that he was trying to hire an NMR manager and that sort of thing. And people came in, and I guess they didn't want to do it or something, because eventually he asked me whether I wanted to do it. And I was a graduate student at the time, but I said sure. So I didn't know what else to do. So I decided that I would become the manager of the NMR facility as well. And then in 1980, John wanted to go on sabbatical to France. And fortunately for me, the dean and the provost both didn't like the idea that I was a graduate student and the manager of the facility at the same time. So they sort of told John that I had to get my PhD before he went to France. So I probably still wouldn't have it if I hadn't been able to do this. But once I did that, he was stuck with me, and I followed him to Wisconsin, and I spent almost 50 years with John, and it's been a great time. Basically, been able to do a lot of good stuff. Anyway, so John now is probably going to present a different history of NMRFAM and BMRB, and he was at Wisconsin from 1984 until 2020. And he started up NMRFAM and BMRB, the Biomagnet Res Bank. And just looking at his publication count, he's done a lot of work. So with that, I think, John, do you want to take over? Hello?
You have to unmute me.
You're unmuted.
Can you hear me now?
Yeah.
Okay. And I'm trying to share. You have to let me share.
Oh, yeah. I thought you...
My share button is
John,
gray.
Well, I'll make you presenter. That's the automatic turn the share button.
Good.
Yeah. Okay.
Let's see.
Yeah, Chinn, I'll let you take care of that if you don't mind. That's just.
Well, I don't mind the play around.
Okay. Now, share. Click on the screen, but it's not coming up. Try that.
Up. It is. Wait. Yes, it's up.
Okay, good. So when I was a graduate student in the 1960s, our lab first had a 60 megahertz NMR, and then we graduated to 100 megahertz. The first superconducting spectrometers were the Varian 220s, and the first ones were only in industry. One was at DuPont and one was at Bell Labs. And I got Bob Schulman's permission to let me collect 220 data on my selectively deuterated protein. And the improvement between 100 and 220 was really amazing. Also, at that time, our lab invited Richard Ernst, who was at Varian, to present a seminar. And in his talk and over dinner, he extolled the virtues of Fourier transform NMR, which he had developed. And so this sparked my dream of pursuing high-field Fourier transform NMR studies of stable isotope labeled proteins. And I sort of wrote up this dream as a postdoctoral fellowship application, which luckily was funded. Fourier transform was not yet commercialized, and the only academic lab that had homemade Fourier transform gear was Mel Klein's lab at UC Berkeley. And in addition, they were about to acquire their own 220 megahertz spectrometer. So I learned Fourier transform there. I learned it on a 60 megahertz spectrometer because they never got it onto the 220 while it was still in the lab. Jobs were tight in 2001 when I started looking for a real position. I heard about only 2 positions. The one at Oregon was filled before I got my application in. The other one was at Purdue, where I was hired and got a $5,000 startup package and got to scrounge glassware from a teaching lab that was turned over to me as my own lab. We had access to the departmental variant HA100 NMR spectrometer. It had a Fabritech computer, and Bob Santini was modifying it for Fourier transform NMR. They promised me a high field NMR spectrometer within 2 years, but didn't get it in writing. And after 2 years at Purdue, the 100 megahertz machine was limiting our research, and the purchase of the Supercon was out of the question. So I began searching for a solution, and the place to look was at the NIH. I was aware of this program from NCRR that supported facilities around the US. And all of these early facilities that are listed here factor into our story. The Stable Isotopes Lab, Los Alamos, they were separating isotopes and also doing some biological studies. And I stopped at Los Alamos Lab on my way from Berkeley to Purdue to learn how they were growing algae on C13 carbon dioxide. I later replicated this at Purdue, and we used the approach to prepare carbon-13 labeled proteins. This mass spec facility at Purdue was long gone before I arrived there, because Fred McLaughlin had moved to Cornell, but it figures in, as you'll see later. The UCSF facility was developing software. They developed Sparky, which we used widely at NMRFAM, and when they decided to stop its development, NMRFAM took it over. The Carnegie Mellon NMR facility was relatively close to Purdue and had excellent instrumentation. The MIT lab figures in because one of its products is Chad Rienstra, who has taken over NMRFAM. Incidentally, I later served on the advisory committees for all of these facilities, of course, except the mass spectrometry facility. Amazingly, Purdue had its own airport at that time with commercial service, and they had a nonstop jet service from Purdue Airport to Pittsburgh, which was perfect for commuting to CMU. I could leave my lab and take advantage of the free parking at the airport and be at the lab at Carnegie Mellon in less than 2 hours. The people who developed that facility were Axel Botherby, the PI, and Joe Dadock, who developed the instrumentation. Joe is an amazing guy. He's considered to be the father of NMR spectroscopy in the Czech Republic or Czechoslovakia. He developed NMR spectrometers in the 50s, and they were commercialized by a company. It's a different Tesla company in Czechoslovakia. He and his family moved to spend some time at Carnegie Mellon, but they got trapped there when the Russians invaded. More recently, the NMR Center was set up in the Czech Republic, bears his name. So I had a phone conversation with Joe a few days ago. He's 95 years old and he's living in Bloomington, Indiana, where his son is an emeritus math professor. So this is the instrumentation I started using in 1974. The Achilles heel of the spectrometer was the frequency generator. it was set by switch buttons, and you sometimes need to give it a whack to get the contacts going. It's amazing, this 220 megahertz NMR spectrometer. They used correlation spectroscopy, which they developed, and the data were processed on the Sigma 5 computer. It was a computer that actually came out of the mass spec facility at Purdue. So apparently went back to the NIH, and then I guess Joe and Axel held up their hands and got a hold of the Sigma 5 computer. Computer is currently in the Computer History Museum. So these are correlation spectra. They were obtained by quickly scanning through the low field region. This generates wiggle beats from every signal, and these can be removed by post-processing by convoluting the spectrum against artificial wiggle beat spectrum of a signal matching the line width of the sample. This technique avoided the water peak and gave excellent sensitivity. The line shape and dispersion was great, and so this is used to follow the deuterium exchange of these cysteine residues, and then we assign them by comparing the HD exchange rates with the HD exchange rates from the literature. 2 years after my promotion, we finally had a high field NMR spectrometer at Purdue. I have put in my own grant application to NCRR, and we were funded for 200 megahertz and a 470 megahertz spectrometer. We were trying for 500, but didn't make it. And in the interim, we first had a 150, was upgraded to 200, and then the 360, which is upgraded to 470. Jerry Dallas was hired from UC Davis as the operations manager, and we're up and running. Here are some pictures of the facility. These are the engineers bringing it together. Bob Santini from Purdue, late Woody Conover and Boris Samsonov. This is the 150 spectrometer, and then here's the later 220 megahertz NMR. As Milo mentioned, when I got a fellowship to support a sabbatical in France, we faced a crisis because Milo was operation manager, and they wouldn't let me go until he got a PhD. So Milo's thesis was a rush job, but he's done a great job ever since. Here's some folks in the lab at that time. Eldon Ulrich, we'll hear more about later. Here's Milo, looking pretty much the way he does now. We were doing carbon detected proton carbon correlations with the proteins that we had isolated from cyanobacteria grown on 13CO2, this technology from Los Alamos. At that time, higher field NMR spectrometers were becoming available with more advanced consoles, including proton detection, inverse detection gear. The problem at Purdue was that the administration refused to provide matching funds for instrumentation. Our facility was fully funded by the NIH. It didn't require any matching funds, but times had changed and matching funds were mandatory. University of Wisconsin-Madison, which was recruiting me, offered generous matching money. So we made the decision to move to Madison. Our first spectrometer there was the Spooker 400, delivered in 1986. It lasted for 34 years, and it was just decommissioned a couple of years ago. The goals of NMRFAM were in a tradition of the Carnegie Mellon facility. First of all, to provide state-of-the-art instrumentation and protocols to researchers, especially younger ones, of course, with valid research needs. And secondly, to pursue continued development of the underlying technology for multidimensional, multinuclear NMR spectroscopy of proteins and nucleic acids. We later added NMR-based metabolomics to the mix, and we'll hear people talking about that later. The other attraction at Wisconsin was the lake. So this is the group in 1987. Ed Newberry was the instrumentation director, and of course Milo was operations director. NMRFAM had this second 600 in the US. The first one went to the NIH. This is Brian Stockman observing a quench of this 600. It survived the quench. It was fine. So at that time, we started doing carbon-carbon correlations in proteins that were labeled with C13 to a level of about 15 to 20% C13. And it really showed how you could trace out the different amino acid side chains and very quickly identify which residue was which. So we continued our carbon-carbon correlations in spectra, and also carried out 2D precursors to 3D triple resonance experiments with proteins that are labeled with C13 and N15. This is a flavonoxin study of Brian Stockman. So this is the lab. Later on, Milo and Eldon and Ed. Mark Anderson took over as the instrumentation director when Ed Newberry retired. And then Mark has retired, and Paulo has taken over from him. We got 900 megahertz NMR spectrometer in 2003, and our most recent NMR lab was designed with a hatch in the ceiling for delivery of magnets. So this is the magnet going in. This is the assembled system, and it's undergoing a test of the safety system, which included a quench. And this is the mature setup for NMRFAM with the 900, 800, 750, 600, and another 600 and two 500s at the time that I left. The final iteration of NMRFAM was a helium recovery system. And recognizing my impending retirement, the biochemistry department wisely hired Katie Hensler-Wildman, who began to take over responsibilities at NMRFAM, and she was responsible for getting this set up along with Paulo. And later, the department recruited Chad Greenstrom. So we had the future in the title. I don't have much to say about the future, except it looks to be bright. NMRFAM is under the able management of Katie, Chad, and Sam. It's supported by a P41 grant, like the ones that we had. I had to bail out of this because they changed the rules and said that you could only have one of these grants for something like 12 years, and we'd already had 30. Their grant focuses on sample preparation technology, data collection methods, technology for software to handle complex systems, and applying the technology to collaborative projects and providing service. So just continuing the tradition of NMRFAM. Also, along with University of Georgia and UConn Health, they're part of this network for advanced NMR called NAN, and you'll hear more about that from Art Edison. There were a couple of 1.1 T solid state NMR spectrometers in order, one for Madison, one for Athens. And in Madison, they're developing knowledge bases for biosolids and material science. Moving on to BMRB, I apologize for this slide with no pictures, but time is short, and this encapsulates 36 years of history. So in 1984, Eldon and I started assembling a collection of sequence-specifically assigned chemical shifts of proteins. There were developing quite a few of those at the time, and we published this in a review article. I took a mini sabbatical in 1987 at Osaka University with Professor Kyogoku, and during that time I started thinking about reasoning for putting these data into a publicly accessible database or data bank. And so in 1988, I had a proposal to establish an NMR data bank was funded by the National Library of Medicine at the NIH. It was first funded as a research grant because we were sort of researching how to do this. The next year, we published a paper on some ideas of how this should be done. And later in 1996, we developed a new format, which became the final format called NMR Star. And I'll talk more about that later. Also that year, the Library of Medicine decided to phase out all of their external grants. And, oh, I'm sorry. 1996, this is when our R1 grant was converted to a center grant, P41 grant, the same kind of grant that we had for NMRFAM. And then we had our first scientific advisory board meeting for BMRB. And then 2007, BMRB became a core member of the Worldwide Protein Data Bank. And we'll talk more about that later. And at that time, deposition of chemical shifts became mandatory for NMR structures. So, okay, this 2012 was when the Library of Medicine stopped supporting their external grants. And we searched around, we talked to NSF, DOE, NIH, and nobody wanted to fund BMRB. So the NMR community got together and put together a paper signed by many of the leading people in the field. It was published in Nature's Structural Molecular Biology. And that sort of shook things loose. And a couple of years later, NIGMS agreed to consider our own grant in support of BMRB. And fortunately, that was funded. And in 2018, the BMRB archive became a parallel archive next to the archive for crystallography in the Worldwide Protein Data Bank. And then in 2019, with my retirement on the radar, when we renewed the R01 grant, we brought in Jeff Hoke as a co-PI. And then last year I retired and Jeff Hoke became the PI and head of BMRB. So that's the overview. So believe it or not, this once was the state of the art for data storage and entry. These are IBM punch cards and the punch card reader. When I was a graduate student, we had one of these hooked up to the Varian 100 megahertz, and you could output a NMR spectrum as a stack of IBM cards, and then you could then play around with it digitally on a mainframe computer. You could take your stack of IBM cards. Later, as a postdoc, we use this. We put our NMR spectra on a bag tape, and then we took it up to a mainframe, and then you'd have to use this punch card to create your control cards for running what was going to be done with the spectrum if you wanted to. I was mostly doing relaxation studies, so you had to specify how to process the data. So this IBM punch card data was the initial format for the protein data bank, and it was also sanctioned by CODATA, a strange organization that I could talk more about later, but take up too much time. And the idea was that it was going to facilitate exchange between diverse computer systems because it was a standard. Of course, the limitations were that your information mapped onto this card, and the format was frozen, it wasn't extensible, and it was incompatible with large data sets. So, for example, when X-ray structures of viruses came out, the old PDB format couldn't be used with viruses. Next, we started playing around with using a relational database format for BMRB. And this is a time when there were over 1,000 publications with assigned chemical shifts. And then our final iteration was NMR star. And this got around the problems with the old 80-column format. NMR star, first I learned about it in discussions with Helen Berman. So she was about to take over the protein data bank and move it from Brookhaven to Rutgers. And this star as a self-defining text archival and retrieval format was used by small molecule crystallographers, and it had an associated dictionary called CIF, a crystallographic information file. So the advantages of STAR is it lends itself to precise descriptions of the kind of complex systems and multiple experiments that are used in biomolecular NMR spectroscopy. Accurate, different labeling strategies, dynamic processes, and multiple NMR experiments. And so Eldon Ulrich was instrumental in developing the STAR and SIFT dictionaries for BMRB. NMR STAR became standard at BMRB by 1997. However, the corresponding format at PDB, because of inertia, only became the mandatory standard in 2019, when the old PDB format was phased out. If you want to know more about this, have a look at these publications, most notably the more recent one in 2019. So the components of NMR star is a data definition language, a data dictionary, and a data file. Some examples here. Uses a controlled vocabulary. All items are clearly defined. it's human readable, it's easily extensible when new experiments come out. It's been adopted by many software packages used in NMR. And as I mentioned before, BMRB is a core member of the Worldwide Protein Data Bank. The initial members were the RCB PDB, headed by Helen Berman, and now by and then the Protein Data Bank of Japan in Osaka, and the Protein Data Bank in Europe, PDBE. BMRB joined, and along with this sister organization in Japan called PDBJ BMRB in Osaka. And last year, the Electron Microscopy Data Bank joined. Of course, they're going to be taking over most of the space in the data bank. China and India are making plans to join. So just a couple of acknowledgement slides. These are all the members that served as the scientific advisory board for BMRB from 1996 to 2020. And Eldon Ulrich, of course, is a major person behind BMRB until his retirement. He retired and then he came back and did some more work for BMRB and then is retired again. This is the late Professor Kyogoku. Our two computer science advisors from Wisconsin. Madison later moved to Greece. After Eldon's retirement, Peter took it over, and these are key people, and now Jeff Hoke has taken it over. So, future of BMRB. It's under the management of Jeff Hoke. John Waddell and Hamid Iqbali are still part of the staff. New staff have been added. They're still funded or underfunded by the NIGMS R01 grant, which has been transferred to Jeff. But they got a supplement for a computer cluster to support CS Rosetta service, which is very popular. And they've had some additional funding from Yukon Health. And they have a new website coming out soon. So thank you for your attention. And I hope I'm within time. I didn't hear the bell going up.
You were 36 seconds short of hearing the bell.
Good.
So if there are any questions, if you could put them in the Q&A. Okay, I guess it's perfectly clear, John.
All right.
Good. Thank you very much.
And I want to thank Pfizer for supporting this, and also for the 2 shots in the booster that I received.
I agree with that. I've had 2 shots in a booster as well. Okay. Sorry?
I was gonna, who's the next? I think is Andy, right?
You're ready? Okay. Okay, so once again, I'm making sure that I can actually start sharing my screen. And at this point, what we've moved on to the next one. Andy Hinck was student 15 out of 45 students that John Markley had. I was number 3, by the way. But Andy he went to Arizona for a while. Now he's a professor in the University of Pittsburgh. And he's going to talk about increasing applicability of NMR for determining structures of small, moderately sized protein complexes. So Andy, if you're ready.
I need to share access, but yeah, I'm ready.
Okay. Chinn, can you?
Yeah, I got it.
I just made you presenter, Andy.
Mm-hmm. Okay, are we good?
Yep.
All right.
Good.
All right. Thanks, Milo, for the inviting me. Thanks to Chinn for the support of the meeting, and thanks to John, for creating a great environment for doing NMR. I, as a starting student, didn't really know much about this, but I thought we were just supposed to get in there and work hard and learn things about proteins, and that's what I did. And again, John just created a great environment for doing that and letting me kind of do it the way I thought was interesting to me. So, and I really appreciate that. So let me start today. So my interest is in structured proteins and how they enable biological function. That's pretty much how I started out. That was my interest. And that's the area that I still work in. I work in studying proteins of the TGF beta family, which is a very extended signaling family in vertebrates. I guess before I get into my talk, I just want to thank the people who perform the work that I'm going to talk about today. It's mainly a postdoc in my lab, Łukasz Witeska, and he got some help from a PhD student in the lab, Stephen White, and a technician, Chang Bian, as well as an undergraduate student, Isabella Conway, and a previous postdoc in the lab, Marcus Hennen. This slide summarizes something that I think is important for people in the NMR field to be cognizant of. People maybe are already aware of this, but if you look at the current content of the protein data bank, NMR structures represents just over 7%. X-ray structures are almost 88%. EM structures are 4.9%. And you can see, actually, if you look at X-ray structures, they're not actually ebbing. X-ray structures the maximum was in 2020, 11,231 structures deposited. And so X-ray is actually just slowly increasing. There's really no ebbing at all in growth of structures by X-ray. EM structures, I think, as probably everybody appreciates, are exponentially increasing. I don't see any ebbing there at all. And, it's possible at some point it could overtake the X-ray structures. NMR, on the other hand, reached its peak of structure output in 2007, where there were almost 1,000. This past year, in 2020, there was under 300 NMR structures deposited in the protein data bank. So this doesn't mean that NMR is somehow a declining method. NMR obviously doesn't just provide structures, it provides information about dynamics, it provides information about intrinsically disordered proteins, it provides information about metabolomics. It's a really very versatile tool. So it's not defined just by structures that are determined. On the other hand, it is true that the number of structures being determined by NMR is decreasing. So I think that's not something necessarily that has to be. I think that there's still a lot of room for NMR to contribute structural information, particularly about protein complexes, about proteins per se, monomeric structures themselves, but being someone interested in protein structures, protein structure function, what I feel is that while, structures alone are valuable, but what's really impactful in terms of increasing mechanistic understanding are the structures of the protein complexes. So that's what I've that's, to me, that's what's important for my field are the structures, but really more importantly, the protein structures, how the proteins interact to enable their functions. Pretty much all proteins interact with something, either ligands or other proteins or nucleic acids. So I think, you know, in the protein data bank, protein structures of protein complexes are definitely underrepresented. So I think that represents an important potential growth area for NMR, but it hasn't been realized. So why do I think that structures of protein complexes are a worthy target? One is that crystallization of protein complexes, crystallization of proteins in general is challenging, and protein complexes in particular are very challenging. So I think that's not an easy problem to solve. And so that, I think, provides an avenue for NMR. Structure determination of protein complexes under 100 kD by cryo-EM at high resolution is extremely challenging. The structures being determined by cryo-EM are of larger species and at lower resolution. So cryo-EM, even though it's an expanding field and there may be technical breakthroughs, is not really at this time providing structures of complexes under 100 kD. And structure prediction of protein complexes remains challenging. And there are several reasons for that, but I think probably a big one is that often when protein complexes are formed, it requires some conformational adjustment of the backbone. And that's sometimes difficult to model and take into account, and has made predicting structures of protein complexes challenging. So I think there's room for NMR. The thing about it is that I think some of the, you know, NMR is complicated and it also has limitations. So one is that, one limitation is that as the sizes get beyond about 30 kD, it becomes difficult to obtain a full set of assignments, both for the backbone and the side chains, even when deuteration is employed. This is due to just the slow tumbling and signal broadening. And also amide-based approaches are sometimes not compatible with the buffer conditions for a stable complex. For example, if your complex is at pH 8 or higher, it can be difficult. So there are challenges to getting structures. It's not an easy thing to, it's a hard thing to get a structure of a protein by NMR, even a moderately sized one. And then doing so for a protein complex where there's 2 or more proteins involved, the size is perhaps larger, approaching 100 kD. That's actually a fairly challenging undertaking. And I would argue that these kind of approaches based on amide-based signal detection and backbone assignment and so forth, structure determination, is not really appropriate for a lot of the protein complexes that are of interest and, in my opinion, worth determining the structure of. So we've been interested in this and trying to use at least the model system that we study, which is the TGF-beta family signaling proteins, to try to see if we can contribute in some way toward providing a more streamlined path toward determining structures of protein complexes that are moderately sized. By that, I would say up to about 100 kilodaltons. So that's what I want to tell you about today. The technology that I'm going to tell you about is not new. It's just essentially it's going to provide my take on how these existing technologies can be used in concert to contribute toward structure determination of protein complexes. So just very briefly here, this is a schematic of TGF-beta signaling pathway. These TGF-beta proteins are secreted signaling proteins. They're homodimers. They require 2 receptors to signal the type 1 and type 2. The TGF-beta 1 and 3 signal by first binding the high affinity receptor, the type 2, TBR2, and then once the type 2 receptor is bound, it recruits the type 1 to form the type 1, type 2 signaling complex. There's a third TGF-beta, TGF-beta 2. It turns out it doesn't bind the type 2 receptor very well at all, about 1,000-fold more weakly than TGF-beta 1 and 3. And so that TGF-beta relies on a co-receptor called beta-glycan. Beta-glycan binds to TGF-beta, and then essentially there's a handoff to the signaling receptors. And so in this context, the beta-glycan is basically functioning as a catalyst to promote the assembly of the type 1, type 2 receptor signaling complex. This is all well established. My lab has been extensively involved in trying to understand the structural basis for the assemblies of these complexes. And a lot of our structures that we've been determined have been done so using X-ray crystallography, and then more recently cryo-EM. The reason is that these complexes are pretty large. So the first one we determined was this TGF-beta type 2 receptor complex, 56 kilodaltons. It was crystallized to 2.2 angstroms. The next that we determined was the same, but including now the type 1 receptor. This is 78 kilodalton complex, six protein chains. That was determined to 3 angstroms using crystallography. More recently, we've determined the structure of the beta-glycan orphan domain. That's this domain here. This is the structure here. It's a 40 kilodalton domain that was determined by crystallography to 2.4 angstroms. And then we were also have in hand a preliminary model of the complex of the orphan domain bound to TGF-beta as well as the type 2 receptor that's shown here. This is currently at about 5 angstroms resolution. This is about a 100 kilodalton complex. One of the complexes that remains is this one here that I've highlighted. That's between this domain here, a beta-glycan called the ZPC domain, and the TGF-beta dimer. And so that's what we're working on now. We've actually tried to crystallize this too, but after screening about 5,000 different crystallization conditions, including a number of custom-made crystallization screens, we were not able to obtain suitable crystals or even hits. And it has to do with not the unstructured nature of these proteins or their moderate binding, but in fact, the really poor solubility of this complex. But in any case, we've been focusing on using NMR as a tool toward this complex and have been working on it for a few years. I'm going to tell you a little bit about that today. One thing I should tell you is that even though the beta-glycan binds 1 per TGF-beta dimer, if you have the isolated ZPC domain, which is the domain that we're working with, and a TGF-beta dimer, it will bind with 2-to-1 stoichiometry. And that's because this domain normally blocks the binding of a second copy of the ZPC domain. But without this domain here, the orphan, then the TGF-beta dimer can bind 2 copies of the ZPC domain. So that's the complex we're dealing with, the two ZPC domains binding to 1 TGF-beta dimer, 66 kilodaltons. Somehow. Okay, so we set about using NMR to determine the structure of the complex. This work was initially done by a postdoc in my lab, Marcos Hennen. Both the TGF-beta 2 dimer and the ZPC domain are very well structurally well-ordered proteins. They both give nice HSQC maps. We've assigned the spectra of both of these proteins. And so we thought, yeah, it might be a little bit challenging to get the structure of this complex. Again, it's two ZPCs per 1 dimer, 66 kilodaltons, but the structures of the proteins are known. We've assigned their backbones, at least in the free state. And we thought we could just then use methyl labeling as a way to overcome the broadening that would otherwise be encountered with using amide signals. And that's what we did. One just technical note here is that rather than using the alpha-keto isovalerate for valine and leucine methyl labeling that leads to labeling of both methyls, we used instead acetolactate as the source of the carbon methyls. And this is nice because it leads to labeling of only one of the 2 methyls of leucine and valine, and it helps to simplify the spectra to some extent over otherwise the spectra with both methyl groups of leucine and valine. So that's what we set out to do, and it actually worked pretty well. So here we're getting really nice methyl spectra of the 66 kilodalton complex. The unbound form here, we're looking at unbound TGF beta or bound, or it's the unbound methyl labeled TGF beta 2 dimer in blue, overlaid with the complex with the ZPC domain in red. You can see we get really good spectra both for not only the unbound but also the bound, and we can identify from the assignments from the unbound form a number of the signals, and we can see nice shifts in a number of these assigned signals. For example, isoleucine 33, which you see here. This signal here, which is isoleucine 92, and a few others. And these actually shift from the unbound to bound state in a slow exchange manner. So this is a relatively tight binding, relatively high affinity complex. And you can see we're getting nice site-specific perturbations in a limited, not all signals, a limited number, some shift to a relatively limited extent. So this is all working actually pretty well. Let's see. We didn't assign all the methyls we were not able to, by transferring the assignments from the unbound form to the bound form, we were not able to assign all the methyls, but we were able to assign a number of them. We saw some additional shifts in some methyls that were unassigned. And so what we did was we took the two methyls that were assigned that were shifting. We noticed they're very close in the structure. That was leucine 101 and isoleucine 33. We noticed that there's several surrounding methyls that were also, we noticed that there were a number of other methyl shifting but not assigned. So we hypothesized that some of these surrounding methyls were the ones that were shifting but not assigned. So we made the so-called dropout mutants, where we convert leucine to isoleucine or valine to isoleucine to assign the methyls. And in fact, our hypothesis was correct. All of these surrounding methyls were the ones that were shifting but not yet assigned. So this all worked out really well, allowed us to identify the binding site clearly for the ZPC domain on this underside region of the fingers. And we then did extensive mutagenesis. I'm not showing all of it here, just some representative data. Here's wild type. This is SPR data that we use to measure the binding. This is wild type. This is the isoleucine 33A mutant. This mutant led to very large shift perturbations. And you can see when we mutated, it indeed leads to a very significant perturbation of the binding affinity. In fact, we can't even measure the binding affinity here, whereas the wild type is about 2 micromolar. Residues that we identified not to be in the binding site when mutated had little to no effects when mutated. NMR was a great tool here, allowed us to get at this interface on TGF beta really unambiguously. And as I said, we were not able to, in spite of really extensive effort for the 66 KD complex, we were not able to get any information from crystallography. So NMR may be slow, but I think it can be reliable. We've also now done the opposite. We've now done methyl mapping on the ZPC side of the interface and made mutants, and also now identified the binding site on ZPC. It turns out to be this region of the protein here. These two residues are critical, isoleucine 162 and asparagine 82. And we see shifts, for example, and we see large shifts in isoleucine 162 as well as some surrounding residues. This loop here, interestingly, in the two crystal structures of this domain that are available, in one of them, this loop was completely, the density was so weak that it couldn't be modeled. In a second structure, the density was quite weak. So it's not clear to me that this loop is actually, what you see here is actually a correct representation of the structure of this loop in solution. In any case, from the weak density, it seems to be a loop that's at least going to be flexible, and therefore maybe malleable and adapt when the protein binds. Okay, so that's what we got from NMR, and it all worked really well. I guess the one thing the important point here is based on these chemical shift perturbations, immunogenesis data, we worked to then determine the structure of the complex using Haddock based on this data. Unfortunately, there was the convergent ensemble was not very well defined. And when we looked at the packing of the side chains in the interface, it was, in my opinion, not very convincing. So I was really not comfortable with any of the complexes that we were seeing based on this NMR data. So even though the NMR data clearly identified the interface area, and even though we've clearly identified key residues for the interaction, the structure of the complex is still, so at least so far, we have not been able to calculate a structure that's really at least convincing to me. So we want to get additional information. And so we considered other possible sources of information, RDCs, PREs, lanthanide-based pseudo-contact shifts. And what we settled on was that probably kind of easy source of information and highly quantitative and source of information to give accurate structure of the complex would be lanthanide-based pseudo-contact shifts. So we got very interested in this. The big picture here is we're trying to come up and working at the system. We're not only trying to get the structure of this complex, we're also trying to think about how we can make NMR more competitive with crystallography and cryo-EM, something that isn't as hard and is more accessible so that larger number of complex structures can be determined. So I think the methyls are a great way. It really extends the applicability, the higher molecular weight. And you can see, I hope, how that was working well for us. But as I said, I think any chemical shift perturbation information that you get by having those assigned methyls in the free and the bound states isn't, and at least in our experience, not enough to really give an accurate structure. So again, the goal here is how can we get some additional accurate information? To do that, again, we were focusing on lanthanide PCSs. We initially focused on these tags, these very nice tags that give very robust PCSs that were developed by Gottfried Otting's lab. These are fairly widely used. You can actually, if you ask Gottfried nicely, he was happy to send these along. And in fact, we got some and tried to put them on our protein, and we had a lot of difficulty. And so one of the tags that he has, this so-called C2 tag, is attached to free thiols in the protein. And we were not able to do this because our protein is actually loaded with disulfide bonds. We tried adding additional cysteine residues to have an odd cysteine for attachment of the tag. And the protein was just we were not able to get any native protein. It just aggregated and interfered with the formation of the other disulfides. So this is not a viable approach. I know it is widely used, but it's not, I would say, generally an applicable method for proteins that already have disulfides. Another approach was to, instead of having a free thiol, was to put in a non-natural amino acid. Gottfried had published on that, the amino acid being para-azotophenylalanine. We did that. We put para-azotophenylalanine into our protein. And the issue that we ran into that is, while conceptually that's, I think, a good idea, it actually didn't work very well because in order to couple the alkyne to the azide in a click chemistry reaction, you actually, you need this azide group to be retained until the point of having native protein. And for us, that was not possible because in order to fold the protein, to incorporate the para-azotophenylalanine, then fold the protein, we actually have to reduce the protein to get it to, so it's fully denatured and so that we can then fold it. And when you reduce the protein, it actually reduces this azide to an amine. And we tried using more Milo reductants, and no matter what we did, this azide always got reduced to an amine. And so we were not in a position where we could have native protein with the azido group present. to couple to our tag. So this was also not a viable approach. So then we turned to these encoded lanthanide binding tags. These were developed in Barbara Imperiali's lab at MIT. They're based on EF hands from calcium binding proteins. And these tags bind lanthanides with quite high affinity, near nanomolar affinity. And you can attach them either to the N terminal or C terminal. And Harold Schwalbe's group showed around 2011 that you can also incorporate them, splice them into loops of proteins if you're careful enough. And so we thought that this was a good route because it was really amenable to the nature of our protein that had a lot of disulfides and had to be refolded. So that's the route we pursued. And it also avoids this kind of chemistry that a lot of biochemists aren't so good at doing. So that's the route we pursued. And and this just provides an overview of the PCS methodology for complex structure determination, just for those of you who are not familiar with it. So you get your tag protein, you add your lanthanide, you then collect this. So this is the protein 1, the protein with the tag. You then collect and measure the PCSs for that protein. And then once you have the measured PCSs for the tag protein, you can then fit that to get the tensor parameters, which we did. You can see here, this is for a model protein that we were working with. I heard you, Milo. And then once you have your tensor parameters, you can then switch the labeling around. So now the partner protein is labeled the same tag protein on the other side, and then measure the PCSs on the partner protein side. And then with the tensor and the measured PCSs on the partner protein side, you can then recapitulate the structure of the complex using Haddock. And so that's what we did. And so this is all established technology. We're just doing that with these particular proteins that I'm showing you here for the validation proteins that we could actually do it in our own lab. And we were, so these are some different LBT tag proteins, loop tagged, N terminal tagged, C terminal tagged. We get nice PCSs in each case. The loop tags give higher PCSs because the tag is more rigid, but we still do get PCSs for the more flexible N and C terminal tags. We can get the tensors for those, and we can then get using those tag proteins with the partner labeled, we can get PCSs on the partner, and then we can use that to recapitulate the structure of the complexes, again, using Haddock. So the technology is working with this lanthanide binding tag strategy, and we're now applying it to the actual system that we're interested in, which is the TGF-beta2 beta-glycan ZPC system. And so this is work that's currently underway. We've made N and C terminal lanthanide binding tagged ZPC. This is the data for the N terminal tagged form of the protein. So the ZPC protein is tagged, bound to terbium, and what we're looking at here are the shifts on TGF-beta2. So you can see we see nice large PCSs on the partner protein. And this is again with a N terminal lanthanide binding tagged ZPC domain. So the terminal tags, even though they're more flexible, can give pretty large PCSs. And so this is where we're at right now. We're now tagging the partner protein, and we'll measure the PCSs in the opposite direction and then calculate the structure of the complex. So overall, what we envision, and we're not there yet, but what we envision is kind of a robust workflow that can be robustly applied to lots of protein complexes up to about 100 kD, where we go through the procedure that I described using lanthanide binding tags. As written, it will require a sample for assignment, backbone assignment, and then a sample for the CSPs, and then the tag sample. So it would actually require quite a few samples, 8 or 9. So it's still fairly burdensome, but I would say perhaps less burdensome than the traditional route of fully assigning the backbones, analyzing the NOEs, and so forth. So I think this is an improved workflow and applicable to higher molecular weight proteins. One thing I should say is we can probably simplify the workflow a bit by taking advantage of other approaches that have been proposed by other groups, where we just prepare an ILV sample and then avoid the backbone assignment and extension to the side chains by just using different approaches for assigning the methyls directly. So Prestegard's group has published a paper on this. Kaladimos's group has published a paper, and Veglia's group has also published a paper on this. So we may be able to just go in and make the methyl label protein and assign it directly. And that I think could save some time and effort and increase the workflow. So that's it. Thanks for being patient, Milo, with me and not dinging me again. Happy to take any questions.
I was about ready to ding you.
Okay, yeah. I don't know if I'm to look in the chat or Q&A.
The Q&A. I guess it's perfectly clear.
I think Andre has a question. No. There's a smile.
Andy, I cannot even use the Q&A to ask questions. You can't even type. I have a quick question, just interest in this lanthanide PCS. I try to get your insight about how we can use this to monitor the conformational change. Is there any particular, for me, just anyone want to do things easier as possible. So is there any particular comments and suggestions that how we're going to, without directly label too much about your proteins, can you get a free probe in the solutions, the PCS?
I guess you're talking about binding of the lanthanide to
exactly
that are present in the protein.
Yeah.
I'm not sure about lanthanides, but I know that for EPR, that for example, a group here in chemistry, Seth Horn's group, they've been incorporating these like multiple or dihistidine motifs that bind copper quite avidly, and they can then attach their copper that way. To bind lanthanides, histidine's not so good, but it may be possible to do a similar thing where you kind of engineer in some glutamates or spartates that could then bind the lanthanides specifically, and then use that to induce PCSs to detect conformational changes. But so far, I haven't seen that. So far, I've only seen more directed attachment. But I think probably the chemists are interested in that and probably work on that.
Thanks.
Okay, Andy, thank you. And I guess we'll move on.
I have a question from Jacinka.
Oh.
Let's see, I'm going to...
You mean Yasna?
Yeah, I don't know how to pronounce it.
Yasna.
I think Yasna.
Andy, great talk. I wonder if you guys have done, if you have positioned lanthanides to various spots except one in order to determine susceptibility tensors with much better accuracy.
Yeah, absolutely. I mean, we for the model system that I didn't really have time to discuss in much detail, we've done that. So the idea is we're going to use as many of, definitely when you have, using multiple lanthanides with different tensors, the accuracy of the recapitulation of the complex improves significantly. So that's the proposed workflow, that it's not just, we're not using just one, having one positioning of the lanthanide and one tensor. We have, in our proposed workflow, up to 3. So one on the N terminal, one on the C terminal, and one in a loop. Maybe it's not possible to get good data for all 3 for each protein that's studied, but that's the idea, is to get up to at least 3. Of course, you could always make more, but it's just more work making more samples. So it's really kind of, as many loops as you have, I would say, is where you can splice into would be the limiting factor. But so far, even the small proteins that we worked on, there's usually at least one good loop that we can splice into that's not in the binding site. Yeah, so that's the idea, is to make it robust. We want an accurate structure, so the complexes, not poor quality structures. And the more data we have, obviously, the better that we can do that.
Okay, well, I think we ought to move on here. So thank you, Andy.
Mm-hmm.
I'm gonna once again try to figure out how to grab onto this thing.
I already made Art and presenter.
Oh,
Art, you should be ready to go.
okay. Well, I'm showing him, showing something here. Hang on.
Oh, sorry.
This is an introduction to Art. So, Art Edison, number 17 of 45. I was number 3, as I said. He went through Florida, and now he's in Georgia, and he's part of this consortium between Georgia, UW, and University of Connecticut about having 1.1 gigahertz spectrometers, and they're going to be doing liquids in Georgia and solids in Madison, and then computer stuff is being done in Connecticut. Anyway, so Art, in vivo metabolism by NMR, it's all yours.
All right. Thank you, Milo. Thank you, Chinn, for organizing, and Josh for doing all the stuff that you're doing. Let me see if I can share this. And it's really a pleasure. Do you all see this right now?
Yep.
Okay. It's such a pleasure to see at least names, if not faces, of so many people that I know, and to honor John and in this session. It's great. I really appreciate it. So I'm going to tell you about something a little bit different from proteins. I guess I've always been a little bit of an oddball in the Markley lab in that sense. I even worked on small molecules when I was there for the most part. And I'm going to start by introducing or reminding all of you of Beadle and Tatum. And I do this, so Beadle and Tatum got the Nobel Prize in 1958 for, the thing that we all learn about in textbooks is the one gene, one enzyme hypothesis, which really does their discovery a disservice. And I'm going to just in case you're not familiar with their work, and I highly recommend that you look for a 1941 PNAS paper, which was essentially the Nobel Prize winning work, to read about this in more detail. It's really cool stuff, because they basically started all of molecular biology and modern biochemistry. So they worked on an organism called Neurospora crassa, and I'm going to be telling you about experiments on that same organism throughout this talk. They took wild type Neurospora, bombarded it with X-rays to induce mutations. Then they crossed this with organisms of the opposite sex. They can mate it, and then they can collect mutants or genotypes. And if it grows in complete media, then they're interested in it. If it didn't grow at all, then something happened that they're not interested in. So they take a particular mutant, transfer it to minimal media. If it does not grow, then what they do next is that they transfer this to different supplements. So in one case, in this drawing that Beadle made, they add vitamins, and they get the same kind of phenotype as if they had complete media. They looked at aromatic amino acids. They really looked at a whole bunch of different compounds, but they could substitute in the chemical, the metabolite that was missing in this mutation. And through this, it was the first demonstration, the first ability to use genetics to access something that would otherwise be a lethal mutation. So we take a lot of inspiration from Beadle and Tatum experiments, and I'll come back to them in a little bit. But I want to switch gears to metabolomics a little bit now, and because there's a lot that we can do with metabolomics. It's an important part of systems biology, and this is the sort of figure that you can see in many instances where you basically take background natural genetic variation. You can consider the epigenetics, transcriptomics, proteomics, metabolomics, and they all lead down to phenotypic variation, like do they grow or not grow on a particular nutrient. And all of this is influenced by the environment, and there's feedback loops on every level. So we like to think that metabolites are closest to the phenotype. They're the end result of all of this cascade of information, and they feed back on every level. So metabolomics these days is incredibly popular. It's a growing field indicated by this sort of purple in the bottom. This includes also metabolomics, lipidomics, glycomics. This is both NMR and mass spectrometry. So mass spec is I guess I'll say it, that mass spec is the X-ray crystallography for metabolomics world. We're always worried about what mass spec is doing, but NMR is still growing rather consistently as well. All of these fields have growth, but there's a big interest in this. And I also want to just highlight a review article that we published in a consortium during this last year on quantum chemistry calculations for metabolomics. I got my interest in graduate school in doing quantum chemical calculations with John and Frank Weinhold and Milo, and I'm still interested in this. And metabolomics is a pretty interesting kind of area I think that there's a lot of growth in the future for quantum calculations to help us in that area. So I want to give a quick assessment about how we are doing as a field. I know that not everybody here is working on metabolomics, so I'm just going to assign it my grades. So here's an NMR spectrum of urine. You can collect mass spectra of urine or a lot of different biofluids. We can measure a lot of things, and we can do it pretty reproducibly, pretty well, and I'll give it an A-. We're pretty good at the analytical chemistry parts of metabolomics. We can even take information, like this is from a figure from a paper that our lab published during this last year on renal cell carcinoma patients compared to controls, looking at urine in these patients. And you can differentiate them based on whether they have renal cell carcinoma or not. So you can find biomarkers. We're pretty good at that as a field. There are all sorts of tools to analyze data. They're not quite as good as they should be, but we can pull out a lot of information through data analysis these days. Metabolite identification, kind of not as good as some of these other areas. We still don't know what a lot of the peaks are that we measure in an NMR spectrum. So there's a lot of effort being put into this, but we still really have a lot of missing information in every single experiment that we do. So maybe C-. We're not very good at tissue specificity. Most of what happens, at least in humans, is basically looking at urine or serum, and we don't break metabolism down the way it's actually broken down in the body. It's tough to do metabolomics on all of these tissues, isolated tissues. And then finally, I'm going to give us an F, mostly to introduce the topic of my material in dynamics. But most studies, including, for example, the one that we published recently on kidney cancer, is one time point or a couple of time points, but not a dynamic process. And I think that really is a weakness, because metabolism is a dynamic process, and we just don't, almost nobody is really doing justice to this. So I'm going to tell you about how we are addressing that in my lab. So I'll start with data collection and what we're doing to try to measure metabolism in vivo. And this was all started by this very talented graduate student who's soon to get his PhD in another week, Michael Judge. And he developed something called continuous in vivo metabolism by NMR. We started this because we were interested in looking at circadian rhythm in Neurospora crassa, which and we weren't able to figure it out with taking the individual samples, so we took another tack. So we're using basically a magic angle spinning probe. It's in the CMP, Brooker CMP version, so we can actually measure solids as well. And what we do, oops, sorry. So Michael learned how to just grow Neurospora on the bench. This is him holding a little sample of Neurospora mycelia, and he puts it into an HRMAS rotor. So it's about 50 microliters or so of this material. No extractions, no nothing. And then this is a road that we put a hole in the cap. We learned this trick from Andre Simpson's group, who have done a lot of really cool work in this area. So we can actually get air into this HRMAS rotor in the magnet. There's no sample preparation, no choice of solvent for extraction. And so then we just collect a 1D spectrum, and this isn't anything particularly impressive. You know, I think that all of you have seen a 1D proton NMR spectrum. This is in vivo of this organism. But the nice thing is that we don't have to make choices. So we have lipids, we have, you know, water soluble components, everything. We don't have to make choices about what to look at. But I'm now just going to focus in on this region. And again, every single person on this WebEx will recognize that it's tough to sometimes when you're down in the weeds to make a, you know, to understand, does this mean anything? These tiny little blips down here, are they telling me anything about or is this just noise, or what's going on here? So that's not too impressive. But what Michael did was just turn on the 1D NMR basically continuously as this organism grew in the magnet. And so what we could see as little teeny blips before suddenly now emerge as ridges that grow over time when we follow this for, in this case, it's 12 hours of data taken in the magnet. And so this is just a small little region of Neurospora growing in the magnet. There are a whole bunch of things that we don't know. We can identify common metabolites like are shown here. And as all of you can see too, we get these twisting pH changes along the way that I'll show you, will tell us a lot of information. But you can see how just the little tracks that we see here are individual peaks that are real and changing over time. So we think that this is pretty powerful way of looking at this organism. We also have a great flexibility in signal averaging versus time resolution. So we can either decide to get to put in greater number of scans and take lower time resolution points, or we can just have a fewer number of scans, higher time resolution. Then we can fix it afterwards. We can do signal averaging afterwards if we need to improve at the expense of time resolution, so we can get signal to noise built up in processing. Oops. So this is what so in our initial publication, Michael compared samples of Neurospora crassa wild type that were grown under aerobic on the top versus anaerobic conditions on the bottom. And I'm just going to highlight the aromatic and aliphatic regions right here. So here are aromatic region. The aromatic region, you can see more of the detail in here, lots of stuff going on, lots of little peaks coming up. We don't know what a lot of these peaks are still, but you can see differences just at a glance between the anaerobic on the bottom and the aerobic on the top. So aerobic basically means there's a hole in the cap. And then I keep on losing this thing. There we go. And then the aliphatic region, again, you can see differences. So here, alanine is a lot bigger in the aerobic condition and smaller in the anaerobic. Lactate shows up a lot more above than below. Lots of differences when you start to look at the nitty-gritty detail in here. So after in this case 11 or 12 hours, the question is, so how did that organism do in the HRMAS rotor for such a long time? And it turns out that it does just fine. It shows Neurospora has a circadian cycle, and Michael can just take the organism, put it into a race tube, and it has a circadian rhythm right off the bat. So it didn't seem to be harmed in any way in these experiments. So we could collect, this is just a summary of our initial publication on this, where we can collect, this is just a highlight of some of the compounds that we identified in primary metabolism. And I'm showing in red 3 replicates of aerobic. These are just independent runs of aerobic condition, 3 independent runs in the blue of anaerobic. And you can see the general reproducibility of all of these traces. So these are just traces of the intensity over time. Time is on the horizontal axis and the intensity of the peaks are on the vertical axis. And so we can get a lot of information about metabolism in this experiment. We can also, as I pointed out, we see pH dependent shifts. And as all of you know, certain peaks in NMR spectra will move with pH, so we can see glutamate, succinate, formate. They're all telling us the same thing, it turns out, and no surprise, but we can accurately measure the pH with all of this. And we did a pH titration of citrate just to make sure that we were doing everything right. And indeed, we can just map different time points to different exact pH measurements inside the organism over time, and it goes from higher to lower, back up to higher in these experiments. It turns out the switch is when the glucose runs out. We can also isotopically label. So in this case, this is that we interspersed 1D HSQC experiments with proton NMR experiments, just a nosy PR, and this is just showing label with C13 glucose uniformly labeled, and you can see a lot of detail. There are actually other metabolites that'll come up. But what became most useful to us is actually just looking at the non-carbon decoupled proton spectra with the carbon 13 metabolites. So this is glucose. So this is the anomeric proton. These are the C13 peaks on the outside, but we can also see the C12 labeled glucose in the middle. And it's actually behaving very differently. So we can see the C13 ethanol peaks on the outside, and then the C12 ethanol in the middle. And again, it behaves very differently. And so this is summarized right here, where probably the easiest thing to look at is on the right, where I scale the concentration of these different species. So the sort of orange yellow is the C13 glucose. It just goes straight down. C12 glucose actually increases and then goes back down. We can see C13 ethanol cruising up and really in correspondence to the glucose going down, and then C12 ethanol going up more gradually. Eventually they meet the same. So this organism, what I didn't tell you was it was in a starved condition when we started the experiment, and we think that what we're seeing is just breakdown of glycogen in the initial steps, and we saw the glycogen corresponded to that was not labeled, and then we could see the C13 pool going independently. Let's see. Okay, now I'm going to tell you the rest of the talk is going to be on Neurospora, but we also have applied the same technology to leukemia cells. We did a study with a guy named Takahiro Ito a few years ago, just after I got to Georgia. Takahiro had been studying chronic myeloid leukemia, and he had the observation that BCAT1, branched chain aminotransferase 1, is increased in myeloid leukemia cells, but he also had an increase in branched chain amino acids. And the dogma at the time, or it still is predominantly, is that BCAT1 would break down a branched chain amino acid into its keto acid. And so the people at Nature said that they didn't understand what was going on. The reviewers said, prove what's going on. So we spent months working out the details with conventional labeling. We would either put in isotopic labeled KIV or isotopic labeled valine and watch it go, and then incubate it for different times in the cells. And in every case, we would only end up with valine. So it definitely went in the direction that was consistent with, so it went backwards from conventional wisdom. But this took dozens of samples, many dozens of samples and many months of work to complete this. But what we did then when we had the CIVM NMR, concept, we took the same leukemia cells after the fact, and we just incubated them. And in this case, this experiment on the left is with C13 KIV incubated with C12 valine. And you can just watch it, watch the KIV go away. You can see the valine come in real time. This took about an hour. And then if we put in the opposite labeling, C13 valine, it doesn't change. It doesn't go back to KIV. So it's a really nice way to actually screen conditions and look at different varieties of this experiment. So now we have this interesting data. Turns out there really were no great tools to do analysis on this, and so I'm going to walk you through some of the tools that we've been developing recently to try to improve the analysis. The first thing is ridge tracking. So we found that it makes a lot more sense to think of this as a 3D surface rather than just a series of 2D or 1D spectra. And this very talented graduate student, Yue Wu, realized that computer vision, the field of computer vision, had solved the problem of being able to identify ridges on surfaces using HK curvature. So you can find ridge points and discriminate them from these other types of structures. So we call this RT extract, and what UA does, this is the same aerobic neurospora data that I had showed you before. I'm just going to expand in on one little region. So this is just one small peak in this spectrum, and I'll show you now sort of the general idea. You can look at the paper for the real details, but so this is just a trace through this spectral region, small little window of points in here. And then UA can, if he finds that H is less than 0, K is less than 1, then that defines a ridge point. He can find local maxima, connect all of these things into ridges. And then basically there has to be a little bit of manual refinement to clean it up and find the ridges. But all of this can be then just converted into a peak intensity over time, and also the change in the chemical shift over time for every single peak. And we can do this semi-automatically on entire data sets now. So the question then becomes, what do we do with all these ridges? Well, this is just a demonstration that some of some in the surfaces that we published on initially, there were some areas indicated by the asterisks that we couldn't analyze it all before, and UA can now pick them out, so it really helps us. So what do we do with these things? And what UA now is working on is using something called functional principal component analysis, so very much like regular PCA, but these are now the ridges that are extracted from a single CIVM experiment, and you get principal component breakdown of these ridges just like in a PCA. But what we find is that the compounds that cluster on the left side, for example, are consumed in the experiment. So like all of these points here are glucose peaks, and they're all consumed somewhat linearly during that experiment. On the opposite side of PC1, on the positive side of PC1, the compounds are growing, like uridine here, or tyrosine, or ethanol. And so they're all mapping over here. The ones on the bottom show a curvature like this unknown peak at 5.4, and alanine starts to curve a little bit down, and then up above, it's even a different kind of curvature, like choline, for example. So we can see what the eigenfunctions are for each of these things to understand what they're telling us in terms of the behavior of these peaks. We can also make correlation spectroscopy. So taking all of these ridges together, then we can make ridge correlation maps. This is, if you're familiar with metabolomics, this is basically doing a Stocksy statistical correlation spectroscopy that Nicholson's lab developed on these ridges. And UA can do this, and he's gotten about 30 different clusters that all cluster together. They're actually a mix of some of them are the same molecule and some of them are in the same pathway. We've only annotated about 7 of these clusters, but you can see the kind of detail mapping some of this is an arginine cluster and mapping it back to to arginine, and then we're using Gizmo as the reference database for to compare our results. Let's see, keep on losing control of this. Then finally, and I'll transition in the last little bit to new applications, but finally, there's this great paper from 2019 that by this group right down at the bottom, Pfizer and all, that they published something called causal kinetics. And essentially, this is a technique to take time course data and use it to find the most natural, stable structure that describes that data. So it's doing sort of like a kinetic model, but not using it's finding the structure, the topology of the system. And I'll just show you what UA is doing with this. So again, these are just taking the ridges that we get from RT extract from these experiments. So above are the ridges. The intensity is a function of time. We can look at the first derivative as a function of time. You can see that has a lot of different sort of information. But then putting it through causal kinetics, now we have a relationship between all of these peaks, known and unknown, between different groups. And we're really, this is sort of the stage that we're at right now. We still, there's a lot less that we know than what we do know, and we're trying to figure out how these are all related together, and we're trying to map it back onto more conventional metabolic pathways, which is challenging. So in the last bit, I'm going to circle back to mutants and more along the lines of what Beadle and Tatum were interested in. And at the University of Georgia, there were investigators, Mary Case, Norman Giles, and others, genetically, they worked on neurospora for years, and they genetically characterized something called the quinic acid gene cluster during the 1970s and 80s. That they found about 50 genes that were involved in utilization of quinic acid from plants. They're able to come up with the entire pathway, except there's one gene that's never been annotated, QAX, and it's computationally annotated as an inositol monophosphate, but it remains uncharacterized experimentally. So briefly, quinic acid comes in, and it can either go into catabolism through the TCA cycle, or it connects the aromatic biosynthesis. That's the general lay of the land with this. So this is just a QAX mutant now, where Michael, and I heard the bell, Milo, so I'll be just a second. Where Michael is doing some quality control, but I want to illustrate a couple of things. So the blue trace at the start is just the media. The red is quinic acid. The big piece of the quinic acid are here. There's some little impurities in the middle here that don't do anything. And then he lets the whole thing run without quinic acid in black. Actually, and then he runs it with, he spikes in quinic acid in the pink in this case, and then he can take that same organism out and rinse out and replace the media with fresh quinic acid, and that's replayed in green. So basically, this shows a number of things. First of all, that we can replay metabolism. Basically, if you look at all of these details, they just show up the same patterns gone through a second time. But we can also recognize what peaks changed at that. We can recognize extracellular versus intracellular metabolites, which is really helpful. So Michael is doing this on wild type versus QAX mutants, and I'm just going to highlight a few of these differences. So on the top is wild type, and on the bottom is QAX. These are feeding with quinic acid, and I'm just going to highlight a few of these just to show you the sort of detail. So wild type, both of these things will accumulate ethanol during a starvation stage, and then wild type will start consuming ethanol. QAX will start to make ethanol for a while still with quinic acid. This is another region. Choline has very different dynamics going on. There's a 3.2 ppm that we have no idea right now what it is. Very different between the two. I'm going to make sure I go through this quickly. Succinate, very different. So succinate starts to accumulate in the starved state in wild type. It doesn't in the QAX. Then they have similar behavior of use of it afterwards. And then finally, in the more in the sugar and aromatic region, there the wild type up above, it's not doing much in the sugar region. The QAX accumulates a good proportion of glucose 1-phosphate, and then it decreases in a sugar that we think is ribose accumulates too. So there's a lot of detail in here and a lot of information that we can get. We're still working on this. And I'm just going to close really quickly by giving, you've heard a few times now, this NAN network. And the thing that I especially like about this is that it sort of brings me home. So I started off in John's lab way back in '89, graduated in '93, and NMRFAM didn't look like that when I left, but it became that under John and Katie and Chad. And so then I've been with Jim Prestegard since 2015. What we proposed with Jeff Hoke as the PI and the hub of this thing. So we are making a network of advanced NMR where these facilities are connected. There's going to be a solid state 1.1 gigahertz in Madison, a solution state version in Georgia. And then Jeff through NMR box and the BMRB and will be the hub, and all of the data will be going to Jeff to the hub and ported over to NMR box. We're working on knowledge bases at UGA. We're doing metabolomics and structural biology. John mentioned biosolids and materials at Wisconsin. We're having workshops, protocol standardization, et cetera, et cetera. And eventually, we want to do a phase 2 once the phase 1 is established and the computational capabilities are there, and add other facilities into this and make it easier for users to find resources and to get the NMR capabilities that they want. And so finally, I want to thank all of the people in my group that did this work, especially the Neurospora team over here with Jonathan Arnold, our collaborator in genetics, the NAN team over here, and all of you. And then I want to, here's a photo that John and Kainosho and I had just before the shutdown. I was an advisor to the WWPDB through the BMRB, and went to Osaka and had a great time just before pandemic hit. And it's been great over the last several years, John, to be able to see you more and more through these kind of occasions. And so I hope that we get together again soon. So with that, I will stop, and I will try to figure out how to stop sharing the screen. There we go.
Okay, thanks, Art. Anybody have any questions? Please put them in the Q&A.
I noticed that John never answered the question that I had about what happened to the algae tank that he told us about, but I won't go there.
It's a big mystery.
It's a mystery.
Yeah.
Okay.
Yeah, we think that the person in charge of the lab, the growth facility, trashed it.
Yeah, you know, good people are hard to find. You know, it was back then too. It's really tough to find reliable, good people. Tough luck.
I think it was stored in the
He didn't get tenure. So.
Yeah, that's true. That's true. It was stored in the fermentation lab, and then the guy didn't get tenure.
Well, the initial downfall, I won't go into that, but we don't have to air all of our dirty laundry here.
Let's see. We do have a couple of questions in the chat. One is, what is the detection limit for the metabolites in vivo?
Good question. We in the Frontiers paper we published it. It's roughly actually not that bad. It's like we estimated between, I think it's around 40 or 50 micromolar under the conditions that we're running. So it's not terrible. It's a pretty good probe. This HRMAS, the CMP Brooker probe.
Let's see. Thanks. We've got one more. Can you get more information by performing 2D experiments and for your in situ studies?
Yeah, but at a lot of expensive time. And I mean, we could, in principle, do well. We've actually worked on collecting JRES experiments, 2D JRES in real time. We annotate everything with 2D, so we take out the sample at the end of the run, and then we'll extract it and do conventional HSQC and HSQC toxi and toxi. We usually go to Colmar, Raphael Birschweiler's Colmar for that. So that's how we get most of our annotations. But I think that the 1D is really useful, and carbon tracing is really useful, and we can now pick ridges apart. So it's still pretty useful.
Okay, well, thanks, Art.
Okay,
And we'll move on at this point. Okay.
Hello, is the audio working now?
Audio is working, Ian. Yes. Okay. So can you see this is what's going on. Ian Lewis, number 37 of 45, one of the later ones, but he's an associate professor, and whatever this Alberta Innovates chair is, I'm not sure what that means, Ian. Maybe you can explain it. It's University of Calgary. And he'll talk about more metabolism stuff. Ian, feel free.
Thank you, sir. I'm very grateful. And if I can be given permission to share, I will share some content. Oh, here we go.
Oh, good.
Here we go. All right, PowerPoint. All right. And I just need to get this. Okay, is the presentation showing up okay, Milo?
Yes.
Thank you, sir. Okay, well, without further ado, I'm going to talk a little bit about how we've some of the projects that started here and John's group and at the NMRFAM have grown over a number of years and have become the things that we're thinking about right now. So this is our nice, beautiful downtown, and we're joining you live though from Philadelphia right now in ASMS. So we're living in the strange world of Zoom. So as requested, we've put together a little bit of a recollection of some of the starting points here of this story that I'm going to tell you about. And so the genesis of this whole project started when I was working with John here in the NMRFAM. Fortunately, right after they got the 900, so we're very proud, and we had accumulated various herds of undergrads who worked with me on trying to figure out how to wrangle these instruments as metabolomics and as tools. You see that we very unwisely thought the baseball caps and t-shirts were cool at that time. I assure you they are. I think one of the most memorable events, though, was the day that this amazing NMR facility flooded, because at the base...
I was on vacation.
It was a good day to miss. They very wisely put the intake vents for the building at the base of the loading dock, and we got this 3 inches of rain in an hour or something like that. It flooded, so we had the entire NMR facility was a lake. I really unwisely missed the opportunity of bringing my canoe and paddling around in NMRFAM, but so this is how I spent my day running around. And so as you can imagine, John, his facility was underwater, literally, and being the totally unflappable guy that he is, I went up to talk to him, said, oh geez, I was so worried. What were we going to do? Magnets are sitting in water. Oops, hey, where'd my picture of him go? It disappeared. Anyway, so John's reaction was like, okay, And he went back to paperwork, totally unfazed by this catastrophe in the basement. So what he had asked me to do when I arrived is they had recently gotten this grant with Michael Sussman to identify isotope or to harness NMR technology and isotope labeling technologies in order to comprehensively identify and quantify metabolites in these very complex mixtures. And at the time, we really didn't know what we were doing. So my job as a PhD student was, we acquired this beautiful NMR spectrum here, 900 megahertz spectrum here of Arabidopsis extracts. And John's task for me was to assign this spectrum. It was like, so I wandered down to Milo and asked him, so how do I do this? He says, I don't know. It's your problem. Or something to that effect. That's how I interpreted it, Milo. And so we started this crazy adventure with Milo and Marco and Klaus and Gabriel and Eldon and many others there at NMRFAM to try and figure out how to go about comprehensively identifying and quantifying things in these really complex mixtures. And so the technique that that John had wanted us to use and was clearly the best was this multidimensional NMR approach, where ultimately we settled on HSQCs and toxes as being a great, robust way of identifying things in solution. We had to overcome some problems along the way. One of those is that at the time there were no databases of standards, so we couldn't download a spectrum of glucose under the same conditions. And so we spent a lot of time titrating standards in the basement of NMRFAM for many years there to help create this BMRB library. Yeah, so as it turns out, that approach worked pretty well. And so we spent a long time here with these kinds of things. This is my representation of Arabidopsis. This is what it is in various ratios. And so we developed quantitative methods here for harnessing NMR. And that's become the foundation of my research career, and now how I approach a lot of problems are through lessons I learned in John's lab. So now I'm going to talk to you about how I've been using this in more recent years. And so for frame of reference, when you go to the hospital, we go there with the expectation of getting better, but 1 in 20 of us will end up getting an infection during our stay, and that results in 2 million infections a year, 80,000 deaths. And our ability to control these infections is getting worse every year because the rates and frequencies of antimicrobial resistant microbes are becoming more and more prevalent across the world. And at some time in our foreseeable future, antimicrobial resistance is going to cost our life expectancy. It's going to shorten our life expectancy by 20 years and end up costing $2 trillion a year in economic impact. So these are a problem that if we leave it unresolved, it's going to dwarf COVID by a lot. And so this is a problem that we think about a lot, and we have been working over the last several years to develop resources to help understand how microbes evolve at a population level scale, and how this contributes to antimicrobial resistance. And so I'm going to tell you a little story about this. So the reason I was recruited to Calgary, what attracted me to coming to the University of Calgary, is that it is directly across the street from a diagnostic testing center that serves all of southern Alberta. And what's cool about working in that environment is that we have direct access to samples and data from everybody in the province. And so what we've worked on over the last many years now is developing ways of both tapping into that flow of patient samples that goes through the diagnostic testing system, as well as the associated clinical records for all those patients, to help understand how infectious diseases have changed over the last 20 years. And so this is a very a quick view of how antimicrobial resistance has changed over that period of time. It's work done by Soren Wacker. I'm going to try and acknowledge folks who are doing the various elements of this program here as we go along, but Soren did this fantastic work, and it shows a increasing prevalence of multi-drug resistant E. coli over time. These guys up here are pretty concerning here in the very top corner, because those are the ones that we don't have good options for treating patients with, and they're right now present in Calgary hospitals. So what we do in my program is ask, can we use metabolism as a tool to diagnose, fight infections, and understand how they evolve? Naturally, the problem with this is that people have metabolism too. And so our primary challenge is we have to differentiate metabolism that's done in the person versus metabolism that's done in the host. And so I built a facility specifically for this challenge. So it's we use mass spectrometry, NMR, and we also have this really cool setup where these facilities are directly integrated into clinical microbiology labs and engineering facilities. So we have this biosafety level 2 suite where we can do everything from analyze or collect data on high resolution instruments, or fabricate new devices for doing diagnostic testing, or grow Neisseria or other kind of icky bugs, and we can do that all in one working environment. So our platforms that we use for our analyses still include the same multidimensional NMR tools that I developed under John and Milo's guidance. Yeah, so we're still using these same kinds of approaches to identify and quantify things in these complex mixtures using those multidimensional tools. We have now supplemented those tools. So we use both NMR and also high resolution mass spectrometry. We use very similar kinds of strategies and thought processes for that work that we did in our NMR work as well. And then the third platform that we've added here recently is called APEX, the Alberta Precision Exchange. This is a program that I launched to integrate our working lab into the healthcare system. And so we have labs that are directly embedded into the healthcare diagnostic labs, as well as our own R&D labs. And this program allows us to directly access 20 years of microbiology isolates, access patient records from that whole period of time, and in total collect samples and data from 8% of Canadians at any given time, and leverage that for some very large-scale translational research. So here's how our metabolomics data looks. Thank you, Art, for the outstanding introduction, so I don't have to describe metabolomics. So we call these etograms. Basically, we measure everything that goes into the bacterium and everything that comes out, or everything they eat and everything they poop, in technical scientific terms. And so, here are the various things that are eaten by Staph aureus over a period of time, and here are all the things that are secreted. Now, what's cool about this, you may appreciate, every little one of these tiny dots is a different Staph aureus isolate from a bloodstream infection from Calgary, every single one of those dots. So these are the collective phenotypes of every Staph aureus isolate for something like a 10-year span in Calgary here. So this is the distribution of phenotypes that we've measured across this. And so we're going to go and dig into some of these distributions and tell you why they're distributed in various ways and what they mean. Okay, so first off, what can we do with metabolism? So recently we've completed a long series of projects that have shown that we can robustly and very quickly identify microbes based on those profiles of what's eaten and what's secreted. And we can do it in just a fraction of the time that's required of normal diagnostic tools. So these are examples of microbes that cause about 70% of bloodstream infections, and we can very easily identify them to the species level just based on these metabolic profiles. But what else can we do aside from identify microbes? So currently, when you have a life-threatening infection, what they do is they take blood, they take it to the hospital and they grow it, or these hospital labs. And they grow it, and then they identify which microbe it is and which antibiotics it's susceptible to. And at the end of this testing pipeline, which can take anywhere from 2 to 5 days. They'll tell the attending clinician which antibiotics to use. But what we don't do is we don't make any predictions about how virulent a microbe is. And as our diagnostic testing tools have gotten faster and faster, it's changed the opportunity landscape where we could potentially inform clinicians not just which species it is and which antibiotic it'll respond to, but how aggressive a microbe is. And knowing how aggressive an infection is could have a really dramatic impact on how we treat infections, because microbes, like people, have a really wide range of phenotypes. Some are relatively benign and some are really aggressive with respect to virulence, or how how much damage they cause in their host when they're there. And this is a distribution lytic activity for Staph aureus isolates, real world distribution here. And so the concept that we've been working on is that what if you were able to test for these virulence profiles and then match each of these patients up with some kind of tailored therapy that was matched to the individual level of virulence for each microbe? What would be the value of that? Patients who had very aggressive kinds of infections could get very aggressive kinds of therapy and have a greater likelihood of survival. And people who have more benign infections could get gentler therapies, which have much fewer complications. So this is a really win-win of matching patients with the level of antibiotic therapy. Doing this, so we call this concept precision infection management. So doing this, it has a problem though. You actually have to know what all of the molecular features are that predict patient outcomes, all the microbial molecular features that predict patient outcome, and we don't know that. So we launched this ridiculous project, overly ambitious project to try and figure that out. And so what we've done is we've taken 50,000 clinical isolates of bloodstream infections, and we're collecting the full genome sequences, their full metabolite profiles, and their full proteomics profiles. We're integrating that with both the province-wide antimicrobial prescription data, as well as the individual patient records for each of the patients that have these infections. And then we're using a variety of computational approaches to identify the microbial predictors of outcome. And we're using that to develop new tailored antimicrobial therapy strategies that are matched to virulence. So doing this has been, it's been a learning experience. We have had to figure out how to transform microbiology into a high throughput activity. So here's Tom Ridzak, my lead scientist, lead microbiologist on the project, and he has helped design this system here that now is being run very seamlessly by a whole team of folks in my group, but allows us to grow this 50,000 microbial isolates in these very controlled and reproducible kinds of conditions. So here's a distribution of the biomass produced over this period of time through our automated robotic-based approach for doing these microbial growth and preparation procedures. So we're doing a lot of different kinds of analyses, genomics, proteomics, but today's talk is about metabolomics. We'll talk about the metabolomics. So what we do is we collect the data, and then we have all the various different strains that we analyze, and we include standards, metabolite standards that are prepared to match what we expect to see in the samples, as well as ATCC reference controls on every plate, and that allows us to make sure things are growing normally. Then we capture the data, analyze it, and capture those etogram profiles for everything. This is a little look at what the data looks like. This is 1,000 different Staph aureus isolates. So some of the metabolite profiles, some of the same ones that you heard about from Art a moment ago. Succinate, for example. These bars here are standard curves that we run on every run. This is just to give you an idea of reproducibility across the various batches. This has taken a huge amount of work to get it to this level of analytical reproducibility, thanks to very hard work by Ryan Groves on my team, and then Luis here, who makes these beautiful visualizations. So we've just finished our first cohort of these isolates here, these 4,955 Staph aureus isolates dating back to 2006. This is a distribution of these isolates that were painstakingly pulled out of the freezers by Nadia, who's doing all of our frontline microbiology work for our team. So she grew up and subbed all these isolates, and we did all these analyses. So I'll tell you a little story from this. So Ali Khan has been working with the clinical records here associated with these data, and he noticed that there, in 2006, from these isolates, that we had this surge of infections here on MRSA cases here in this 2016 timeline. And not only did he see this surge, they appeared to be coming primarily from this one hospital, this Foothills Medical Center here. This is one of our 3 hospitals here in the Calgary zone. And you see that we had this surge in infection rates here. And at that same time, we also saw increases in mortality here from that same period of time. So that was reflected here in mortality rates, also here at just that facility, as well as increases in costs and other kinds of metrics tracking for this same period of time. So we collected the full genome sequences of all the isolates here, but from the same spike, we were able to show this is done in partnership with the Broad Institute, Ashley Earle, and Bruce Buren. We showed that, Bruce Walker. We showed that there was this explosion of different sequence types that occurred at this period of time. So this is the entire genetic assembly of all the staph isolates we've analyzed over this cohort, and then from this particular window, we saw this big explosion of different sequence types. And as it turns out, these different sequence types could be lumped into a couple of different groups that were clustering according to their MRSA profiles. So we also showed from these data that there were surprisingly some significant differences between methicillin resistant and methicillin sensitive Staph aureus lines with respect to their metabolism and their metabolic profiles. And some of the most significant differences here were seen among ornithine, arginine, and citrulline. So those of you who know your biochemistry should know that those 3 molecules belong together. We'll have a quiz at the end of this to see who's got it. But yeah, these 3 molecules are from the same pathway here. And so it's pretty exciting and not at all what I expected that we would see this tracking with drug resistance. In fact, I had told Luis that I really didn't think that this was a very high probability target. But as it turns out, there were also really significant differences in protein expression here from those same groups, as we did collect all the proteomics data from these isolates as well quantitatively. Now, not surprising, the gene that encodes for methicillin resistance, this MEK-A, was the most differentially expressed gene amongst the MSSA and methicillin resistant isolates. But also this cluster of arginine catabolism proteins was also some of the most differentially expressed genes. So we found a spike in MRSA. We showed that there were significant metabolic differences attached to this, and we showed that there was also differential expression in things related to the catabolism of arginine. So overall, this is what that little story showed, is that these different isolates here showed significant differences in the expression of this ornithine carbamyl transferase, which is part of the urea cycle. And also there were these significant differences in the associated metabolites from that pathway. So to our surprise, this wasn't the only metabolic phenotype that we found co-segregating with a clinically significant phenotype from the microbe. Oh yeah, as it turns out, this disruption in the urea cycle here, which is what we're showing here, this is the urea cycle, makes some sense. Is there's recently discovery of this ACME element, which is this arginine catabolic mobile element, is it turns out that it's on a gene cluster that is right next to the MECA gene. And so there's some co-selection here for this arginine catabolic change that goes along with methicillin resistance. Now, why this arginine catabolism is being selected along with MECA is still a mystery, but it was clearly being co-selected and highly significant across this 20-year cohort. So, but to our surprise, this wasn't the only thing that was segregating along these, according to their urea cycle activities. So the small colony variants are these smaller growth forms of Staph aureus. So they're still Staph aureus, but they have genetic changes that make them grow smaller. There are a variety of different changes. Usually these are mutations in heme catabolism. But we were also surprised to find that these isolates, these are interesting clinically because they're associated with much worse clinical outcomes. That's why people are interested in these small colony variants. They generally have much worse clinical outcomes. They're frequently associated with infections in joints and this sort of thing. We're surprised to find that there was also a really significant metabolic association with urea cycle activity amongst these clinically associated isolates. Now, in this particular case, the difference here was in arginine deaminase, so a different enzyme, but still the same pathway, and also resulting in these fairly significant changes in urea cycle activity. So two seemingly unrelated pathways, both linked to significant clinical progressions, and both linked to this same metabolic pathway. So this is the very first look at an exciting new story. So some of these data we've only had here for a week or two. So we're really thrilled to finally be digging into this. But I'm going to wrap it up saying that we have just kicked off this really probably overly ambitious effort to map all of the molecular predictors of patient outcome, both metabolically, genomically, proteomically. And, and this little story that I've just showed you shows how we can use these data to track outbreaks, find out which microbes specifically are responsible for the outbreak, identify molecular features that are tracking with the outbreak. Didn't have time to tell you about the whole mechanistic story, but identify the biochemistry behind some of these mechanisms. And at the end, get some targets that we can look for in order to test for virulence in clinical settings. So these are the folks who are doing this significant work, lots and lots of people who are contributing to this. And none of this would be possible without this great team of folks and Genome Canada support. And one other thing I want to leave off on here is that diagnostics gets, I think, a bum rap. It's a utilitarian kind of thing, but it's something that we are in particularly good position to do something about as technology experts. So I really encourage folks from this group to think about the tools that you work on and think about how they can be transitioned into diagnostics, because many of the things that we know offhand can have the potential to affect a lot of people's lives. And so my challenge to you is, are you going to help try and prevent some of these deaths? And of course, I really have to thank Dr. Markley for setting the trajectory for my career and taking a chance on bringing me in. Because I wasn't an ideal pick, and I'm really grateful to you and for starting my lifelong love affair with science and metabolism and showing me what it's all about to be a careful scientist. I'm really grateful. And I'm also really grateful to Milo, who's put up with me for many years and taught me many things that I've forgotten again and again, and he's retaught me too. very, very patiently. I'm really grateful to both of you and the rest of the NMRFAM team. Thanks, guys.
Thank you, Ian. So we ask, you can put some questions into Q&A. Art had a question. Do you routinely measure both the media and intracellular metabolites? Does Does one give more useful information than the other?
Yeah, they give different information, right? So people really are interested in intracellular metabolites. Actually, I'm doing a little sleight of hand here. I'm showing you some intracellular data and some extracellular data. So the extracellular data tell you flux. The boundary fluxes when you're measuring a microbe that's held in a period of time, in a medium for a period of time. So measuring the things that go in and the things that go out tells you about pathway activities. The delta tells you pathway activities. And when you measure intracellular things, unless you use an isotope label, you're measuring steady state. And so there are times when steady state metabolism is useful, but generally we're mostly interested in flux, dynamic changes, like the last point on Art's very excellent slide deck. We're also interested in trying to explore that. And so this is our way of doing that. So there is, you know, for routine kinds of projects, I find extracellular data is usually more helpful. But it really depends on the application. Yeah.
So Ian, just if I could just follow up since I'm unmuted now.
Sure.
That was a bad move, Milo. If you want to be useful to the pathology lab, if you want to get a diagnostic rapidly for an infected person in the hospital, what do you pick? Do you culture it? What do you look at?
Yeah, that's the cool thing about microbiology, and some of this could also be applied to oncology and some of the other things, is that there's already clinical practice to take a sample. It's already clinical practice to grow the microbe in a lab setting. It's already clinical practice to perform some lab tests. So my proposal is, why not add one more test, right? And I think if you were to take that mindset, it's shocking if you walk through a clinical microbiology lab, how outdated, or clinical lab in general, how outdated the technology is. Like MALDI mass spec revolutionized the field in like 2010. MALDI, right? So like they're at least 20 years behind the curve on technology. So when you walk around, with your lens as a tech expert, you'll be shocked what stands between you and a life-saving diagnostic. And so I think many of the things that we work with could be directly integrated, and finding tools like this, to your point, that are already being cultured, or already being interacted with. It's a simple vehicle. You add another test along the way.
There are a couple more questions. How accurate do you think the models might be? Are you also a patient immune response as a predictor?
Yeah. So so part of our complexity is the whole precision infection management is there's a patient attached to it. If it were just microbes, it'd be easier. So that's part of what we're trying to capture here with the patient records, is understanding the patient context, and absolutely, whether somebody has HIV or is elderly, or like the patient status has an immense impact on their trajectory, right? And we have to unravel that from the microbial predictors. And I don't have a really great solution for doing that, other than we're trying to tease out things that are covariant with the microbial traits, and try and isolate those things as best we can.
And the final question, I think, is lots of unknown metabolites that are significant. How are you going to go about identifying those?
Collaborate with Art.
That wasn't Art's question. Dan Rafferty. You can ask Dan.
I mean, we do what we can, but I think that there's no solution to the fractionation, identify, and there's no solution to the hard work part. Yeah.
Okay, thank you, Ian.
We did have another question in the chat. Do you know what makes a strain so virulent?
Yeah, well, no. Some things we know about. There, for example, the production of exotoxins, for example, are things that lyse host cells, or great examples of things that are linked to virulence. But actually, the list of known virulence factors is pretty short, and they don't do a very good job of describing why strains are aggressive or non-aggressive. Like, you certainly can't, exotoxin P doesn't explain all of the virulence of staph. So that's the point of the whole program, is to find out how much of it is tied to microbial growth rate or biomass production or exotoxin production, or things strange that we weren't anticipating ahead of time, like changes in arginine catabolism, right? So that's part of the magic of the story. And I don't know how it's going to evolve, but we're thrilled to have our first full cohort of data to be digging into these questions.
Okay, thank you. I'd like to thank all the speakers today. I mean, it's been great. It's long. Well, I'm glad I extended it to 5 o'clock, because here we are, and it's getting close to 5, or, well, a few minutes. And I just want to thank everybody for showing up and such great talks. It's a lot of fun. It was enjoyable. And I just want to point out that the next Tuesday is going to be another Chicago area NMR discussion group session. And Josh Karutz, who answered a lot of the questions today, is going to be chairing that one. And it's going to be a topic of NMR education and applications across greater Chicagoland. As this is a Chicago area NMR discussion group, however, because I sent out emails around the world, I think we have people from around the world on this one. So it's not quite Chicago area anymore, but I'd like to also thank Chinn and Pfizer for sponsoring this. And I guess at this point, I'll just say goodbye.
I just want a quick mention that this seminar conference is recorded, and we'll ask presenters to their consent to release later.
Thank you.
All right. Thank you all.
Bye.
Bye.
Thank you.
All right. Thanks, everybody.